Okay, welcome to Let's Get to the Marks. We're looking at A-Level Biology Lipids. This is lesson 2. Okay, in A-Level Biology, we split lipids into two main categories. That's triglycerides and phospholipids. Triglycerides are a long-term energy solution. They're a storage molecule that contains lots of bonds which can be released for energy. They're also used for insulation. And then we've got phospholipids. Well, what do phospholipids do? They make up the plasma membrane or the phospholipid bilayer in the cell membrane. And you can see these are the phospholipids circled here. So we're going to look first at the triglycerides and go through everything you need to know for the A-level biology content on triglycerides. Now they contain the elements only, they contain carbon, hydrogen and oxygen. Sound familiar? That's the same as carbohydrates. Now the structure of a triglyceride, it's made from a glycerol molecule. That's a type of alcohol. It's actually propan 1,2,3-triol. But this glycerol molecule is connected by chemical or covalent bonds to three fatty acid molecules. So triglycerides are made of one glycerol molecule and three fatty acid tails or molecules. The fatty acid tails have the same general structure, except for they have something called an R group, which is the bit that varies between them. Now let's look at how they form. So here we have the glycerol, the displayed formula in green, and we have three fatty acids here in yellow. Notice the capital R on the side of the three fatty acids. That's the R group. And that means it can be different lengths of carbon and hydrogen atoms. So you could have 20 on one, 5 on another. Some have got double and single bonds. We'll look at that in a second. So the glycerol molecule reacts with the fatty acids in something called a condensation reaction. And what do you think of when you think of condensation, like water droplets in the shower on the mirror? That's right, water gets removed from the glycerol and the fatty acids. You can see the green H from the glycerol being removed here with the yellow OH from the fatty acid, and that releases a water molecule. So how many water molecules will we need to remove to form one triglyceride? Well, it will be three water molecules because we've got to connect the three fatty acids to the glycerol. And we form this triglyceride down here. You can see it right there. The bond formed from this condensation reaction is known as an ester bond. So ester bonds hold together the fatty acids and the glycerol molecules. So now we're going to look at a little bit of detail to do with the fatty acid side chain. Now the fatty acid tails in a triglyceride can be different. You can either have saturated fatty acid tails, like you can see here. Saturated means the R group, which I've circled here in red, this is the R group or pink, only contains single carbon-carbon bonds. So that's the R group, the bit that can be different. And this one only contains single carbon-carbon bonds. And we call this saturated, a saturated fatty acid. And then we have an unsaturated fatty acid. You can see here that the R group contains a double bond. And unsaturated fatty acids have to have at least one double bond. We call them mono unsaturated fatty acids, but they could have more, 2, 3, 4, 5, however many you want. And then there'd be polyunsaturated fatty acids. So this is a mono unsaturated or just an unsaturated fatty acid. Now let's look a little bit closer at the two different types of bonding. So here at the top we've got the saturated fatty acids. Now when the tails are like that, they can get close to each other between different triglycerides, and this increases the intermolecular forces and gives them high melting points. These are seen as unhealthy and more likely to form fatty deposits in the arteries. You know, they're going to build up and you're going to get plaque and atherosclerosis and potentially a myocardial infarction. You're going to end up with heart disease. Whereas the ones below are unsaturated. Now notice the kind of bend around the double bond, and we call these kinks. Unsaturated fatty acids have lower melting points due to the kinks in the chain formed by the double bonds. What this means is that the tails can't line up closely together because they're all bending over the place, and this lowers the melting point, which means they're more likely to be liquid at room temperature, like olive oil, coconut oil, and other vegetable oils. And these are associated with good heart health, and they're actually good for you, like this olive oil here. Okay, I talked earlier about triglycerides. They're excellent long-term energy storage molecules. Look at all those chemical bonds they contain. These release lots of energy when they're broken down. Now, the technical biological term for breaking down a molecule is hydrolysis and involves water molecules, but they will break those carbon-carbon and carbon-hydrogen bonds and release energy. Now, anyone who's tried to wash oil off of their hands, you'll know it's very difficult because, of course, triglycerides or lipids are insoluble. And this is good because they have no osmotic effect, which means they don't make cells burst. They don't change the water potential of cells, which could lead to cells bursting. Also, triglycerides clump together with the glycerol molecules. They're the little green rectangles here facing outwards. and the yellow hydrophobic tails facing inwards away from the water. So you'll know if you mix oil and water, you can see it form these droplets. Okay, we're going to move on to the second type of lipid. So there's two, the triglycerides we've just gone through. We're now going to look at the phospholipids. And the phospholipids, these lipids are found in cell membranes. And what we're going to look at here is how their structure is different than the triglyceride. So if we look at the phospholipid, it's almost the same as the triglyceride, except for the fact that it's made up of a phosphate group, shown by this blue circle or sphere, and one less fatty acid, only two yellow fatty acid tails. It still has a glycerol molecule, and it still has ester bonds. The phosphate group is attracted to water. We call that hydrophilic or water-loving. And the fatty acid tails make up the tails of the phospholipid, and they're hydrophobic or water-hating. You can see the ester bonds holding the fatty acids and glycerol together. Now, cell membranes have a really important job. They control what enters and what leaves a cell. We can see here I've circled the heads of these hydrophilic heads, which face outwards, and they are water loving. And this allows interaction with the tissue fluid in which the cells of your body are bathed. So the cells in your body are surrounded by tissue fluid, which is mainly water. These hydrophilic heads need to be able to interact, and that's why they're hydrophilic. Now, the hydrophilic heads facing into the cytoplasm also are charged because the phosphate group has a charge, and so they love water. And this is good because we want our cell membrane to be able to interact with the cytoplasm inside the cell. However, the tails of the two layers face inwards and they're hydrophobic. And this is really good because these hydrophobic tails make the membrane impermeable to water loving molecules. Glucose and amino acids cannot just move in and out of cells willy nilly. They actually need special transport mechanisms to get through. However, non-polar molecules that are soluble in oil or lipids can get through. Okay, let's look at the test for lipids. This is the final bit of scientific knowledge you need for biology A level with lipids in biological molecules. So take your sample, add ethanol and shake, then pour into cold water. And if you have any lipid, like this oil here in this tube, you're going to get a milky white emulsion. And it's as simple as that. Okay, guys, I hope you've enjoyed the video. This was on cell membranes. Don't forget, let's get to the marks. Like, subscribe, share, all that jazz. And I'll see you next time.
You're still here? It's over.